And there's a lunch as well. So if you're going to buy lunch that day, you might as well come to a conference. Okay? It's a small conference. It's a local meeting. It's about microscopy and, as it says, microanalysis. Very, very small meeting here in northern Arizona. We're going to have probably 2 or 3 students presenting from my lab on their research projects. We're going to have a graduate student, Robert Biller. He's working on his Ph.D. in my lab. He's giving one of the talks at the podium. There's only 3 talks. Many conferences are like a day or 2 long of talks. This is only basically it's about half a day. From 11:30 is the opening ceremony, and then I think there's a lunch, a buffet lunch right after that, and then they start with the talks and the poster session. Okay, so if you're not planning on leaving for spring break until Saturday or Friday night, this would be a great opportunity for you to see some of the research that's happening right here on campus and in the region. Okay, not a big conference, very low key. Okay, you can probably dress as you are. It's in the ballroom over in the Bois. Okay, and so just throwing it out there for you all.
Are you going to be there?
I will be there. I will be there with bells on, as they say. Okay. What kind of dog do you find in a bakery? Oh, you guys have some ideas. What kind of dog do you find in a bakery? Not a hot dog, that's a good one. Beagle? Ooh, I like beagle. That's good. A purebred. Oh. All right. When do astronauts eat lunch? When do astronauts eat lunch?
When they have space?
When they have space. I knew. At launch time. Come on. I mean, it makes sense, right?
Yeah.
All right. A couple of other announcements. So midterm grades are posted. Midterm grades are posted. The withdrawal deadline was the 13th, which is this Friday. But it's been rescheduled to the 23rd, after spring break. The whole idea is to get, for midterm grades, to give you guys an assessment of where you're at. And also in a large classroom like this, it's an opportunity to find out if we made any errors. And sometimes we make some errors on entering numbers or not entering numbers. Okay? Your exams are here and in the back. I encourage you to pick them up and look them over. I will post the exam online like I do with all of them after the fact as PDFs. There's a folder called exams or old exams or something like that. So if you don't want to have to come in and pick up your packet, you can just look at the packet online. Okay, that resource is available after every exam. I've assigned worksheet 3 and quiz 3. They're not due until the Monday after spring break, which is the 23rd, which also happens to be the withdrawal deadline. Is anybody confused about what withdrawal means? It's not like, I'm going to miss you so much, Dr. Keller, over spring break, I'm going to have withdrawals. Okay, I'll miss you too. Does anybody have a question about withdrawals? So the idea is, if you're not doing well in this class and you think that you may not get the grade that you want, and we're about midway through, you have the opportunity to withdraw from the class, take a W instead of getting an F, for example, or even a D. Okay. It shows up on your transcript. A lot of folks think that it's better than that terrible grade. That is up to you. I cannot tell you what to do. I'm not trying to pressure you into doing. But the university thinks it's important that you have information about what your grade is so that you can make an informed decision before the deadline. It's also why they pushed the deadline back. They didn't push the midterm deadline back. For us, they just gave you a little bit more time to think about it because spring break is next week and you might be leaving and then forget to do something. Okay? So the worksheet is due on the 23rd by 11:59. So that's Monday when you get back. Now, if you're not going to do anything over, there's a couple strategies and a couple of scenarios. You can totally take spring break off. Okay? You have an exam the week after spring break. It's not Monday. Because I've done that in the past and it doesn't go well. So the exam is Wednesday. So some of you will be like, that's plenty of time. I'll get back on Sunday. I'll study Sunday night, Monday, Tuesday, Wednesday morning, because the exam's not till Wednesday afternoon. Maybe that's you. Some of you have 2 exams under your belt, and you're nervous about that midterm grade. I'm going to suggest you study over spring break. Okay? This exam 3 tends to be one of the most difficult exams of the semester.
Why?
Because of the material. It tends to be the most difficult exams of the semester. This one and the next one. Okay? So I hope you take it seriously. The quiz 3 is due the Tuesday, the 24th, after spring break. So you theoretically could take it all off, come back on Sunday, start studying, do your worksheet on Monday, get it in before midnight, do your quiz on Tuesday, get it in before midnight, and be ready for the exam. So that is a possible scenario. Another scenario might be, you know what, I'm not good about remembering, Dr. Keller, so I think I'm going to get my worksheet quiz done before I leave for spring break. You can do that as well. You could get it done and save it. And then if you want to refresh and study over spring break, maybe open it back up and walk through it again. And then double check your answers. And remember, it'll submit automatically on the due date. So that's another more conservative approach. A third scenario might be, I'm going to do it before spring break, so I don't have to worry about it. But I'm going to, you know what, on spring break, for at least 2 or 3 days, for about an hour and a half each, that's not a whole lot of time, that's 4.5 hours spread over 3 days, an hour and a half, I'm going to study. And then I'm going to come back and hit it hard on Sunday and be prepared for the exam. That's probably my favorite scenario. For me to tell you to study every day on spring break, I mean, come on. Okay? But don't blow it off. Here's my goal, is I don't want you to go away from spring break and ruin all the progress that we've made. Okay? I don't want you to come back and forget everything that we've done. Okay? All right. Any questions before we get started?
Did the SIs happen to have a schedule for that period of time before the quiz and after spring break? For like any
they're working on their schedule. They also committed that they're going to have a study guide and a practice exam available before spring break. Okay? For those of you that actually want to take my suggestion and just, I'm not saying waste your whole spring break on studying. I'm just saying don't come back dumber than when you left. Okay? Please. Okay? Let's just be real here. Have a good time, be safe, and, you know, just don't do one of these. Okay? It's kind of, you know, maybe a little bit of that. All right. So we're in neurotransmitters. We're going to wrap up the nervous physiology, and we're going to get into our central nervous system today. And we theoretically have 3 lectures before the exam. We have today's lecture, we have Wednesday, and we have the Monday that we come back. So you'll have a lecture Monday when we come back that kind of helps get you back in the zone before the exam. But there will be some new material. So when we left off last time, we were here. We left off here, and some students have some questions. In fact, in SI, some of you have asked some of the SIs. I'm a little confused about what the sodium potassium ATPase does during this whole time. Like, what's it doing while this is all happening? What's it doing? It's moving ions. It's pumping 3 sodiums out. It's pumping 2 potassiums in. It doesn't turn off. It runs all the time. It's like that leaky faucet that your father hates. It's running all the time. Okay? So, during this action potential, the movement of those ions with those voltage regulated gates is so much bigger that it overshadows what's happening with the sodium-potassium ATPase pump. The sodium-potassium ATPase pump effect is really seen at resting membrane potential before and the return to resting membrane potential after. Remember, its role is to help maintain it. So it's constantly going, and it gets overshadowed during the action potential, but it doesn't turn off. So hopefully that clears that up. We had a different figure that looked like this from our textbook. Same thing. So in the pre-1 phase, where it's resting membrane potential, and then in phase 7 and beyond, where it stays in resting membrane potential, that's when you're really seeing the effects of the sodium potassium ATPase pump.
Does the pump rate change, or is it just constant?
Constant. Constant rate. It will change slightly with metabolism in different phases of life, things like that, but for the most part, it doesn't really move, especially as it relates to this topic. Okay. So, let's talk about at the synapse. We have a presynaptic neuron, and we have a postsynaptic neuron. The synapse is the gap in between. And we have a electrical signal coming down. It converts it to a chemical signal across the synapse, and then it converts it back into an electrical signal on the other side in order to propagate the action potential. These neurotransmitters, they only bind for about a millisecond. That's it. If you don't have a new neurotransmitter to take over from there, then the stimulus will stop. So the way that the stimulus can stop, and one of the questions that leads to this is, why do we even bother having a synapse? Why have a gap? Why not just make the neuron go exactly where you want it to go? Why have these gaps in between? It seems inefficient. Well, you now have the ability to control it or modulate it. So you can influence how long it lasts. You can influence how sustainable it is. You can influence the intensity, how strong the signal is. And when you turn it off in one of 3 ways. Diffusion of the neurotransmitter. Well, it actually escapes out of the synapse and into the extracellular fluid. In the CNS, we would see the astrocytes. Remember, those are the cells, those are glial cells that help to regulate the chemical environment. So they actually absorb it. Or you can have the neurotransmitter reuptaken. The synaptic knob, where it came from, actually reabsorbs it back into vesicles. And you recycle it. Or thirdly, we could actually break it down, or through destruction. There's an enzyme known as acetylcholine esterase. It's abbreviated ACH, it'd be ACH, little h, capital A, capital C, little h, ACH, and then capital E. Acetylcholine esterase. That breaks down acetylcholine into its 2 constitutive parts, choline and acetate. Then the synaptic knob will reabsorb those 2 ingredients in these vesicles, and they'll be able to recycle those parts into making new acetylcholine. Okay? I want to show you a video of what goes on at the synapse. Okay, so this is a video that's describing the effects at the synapse. You can see, here's the presynaptic side, here's the postsynaptic side, and the acetylcholine is dumped out. This one's dopamine, excuse me. Dopamine is dumped out, and as dopamine is dumped out, it binds to its receptor, and its receptor causes a postsynaptic activity. Okay? And you can also see that when the neurotransmitter dopamine is done, it's reuptaken. That's one of our ways of bringing it back in. Well, if we play a little research here, and we click on this button, research is so fun, look how easy this is. We add cocaine to the system. So when cocaine is added, it blocks the uptake of the dopamine neurotransmitter. So with the dopamine neurotransmitter being blocked with respect to uptake, then the dopamine is here to continuously stimulate the postsynaptic side. So this is a pharmacologic example of how a drug, known as cocaine, okay, but there are natural cane derivatives that we have in our body. We have endogenous opioids that do the exact same thing with respect to dopamine. Right? And these endorphins and these enkephalins block pain, just like what cocaine does. So cocaine is a derivative of a natural occurring substance that we actually have, and that's that runner's high, or that sympathetic activity that people fight through the pain, they don't even realize it. Sometimes people have superhuman strength. You've heard of the scenarios of a mom pushing a stroller and like a truck rolls over the stroller and this 105-pound mom picks up the truck and pulls the baby out. And everybody's standing there going, what just happened? Okay? So enkephalins and endorphins effects pharmacologically have the ability to do these kinds of things. There's another activity that I want to talk about, and I want to show you. I want to show you this video that talks about Botox. Okay, so let's watch it.
The presynaptic neuromuscular nerve ending contains membranous vesicles prepared to release its stored neurotransmitter, acetylcholine. Neuronal stimulation initiates a cascade of events that leads to the fusion of the neurotransmitter-containing vesicle with the nerve membrane. This process is facilitated by a group of proteins comprising the snare complex. The membrane fusion results in a release of acetylcholine into the synaptic cleft by a process of exocytosis. The acetylcholine diffuses and eventually binds to receptors on the muscle, leading to muscle contraction. Botox, Botulinum Toxin A, consists of a heavy chain of 100 kilodalton and a light chain of 50 kilodalton, making up the 150 kilodalton core type A molecule. The toxin is protected by accessory hemagglutinin and non-toxic non-hemagglutinin proteins, yielding a uniform 900 kilodalton complex. This illustration shows a cross-section of the spine, with a motor neuron extended.
Oh, bummer. Hang on. Let me figure out what happened there. Maybe I just need to play the file. Sorry, guys.
The presynaptic neuromuscular nerve ending contains membranous vesicles prepared for the nerve membrane by a proxy. Botox, Botulinum Toxin A, consists of a heavy chain of 100 kilodalton and a light chain of 50 kilodalton, making up the 150 kilodalton core type A molecule. The toxin is protected by accessory hemagglutinin and non-toxic non-hemagglutinin proteins, yielding a uniform 900 kilodalton complex. This illustration shows a cross-section of the spine, with a motor neuron extending into the muscle and a sensory neuron extending out of the muscle. After injection of Botox, it would be expected that most of the neurotoxins would remain at the injection site. The Botox core molecule dissociates from the accessory proteins and targets the nerve endings. The binding domain of the Botox core muscle is the C-terminal protein of the heavy chain with an acceptor on the nerve. The Botox core molecule enters the nerve cell by a process of receptor-mediated endocytosis. It is the heavy chain that contains the binding domain. The toxin is now contained in a membranous vesicle inside the cell. Soon after, the light chain is released into the cytoplasm of the nerve terminal,
So where are
it
we?
begins to cleave
With the
one of the
very
snare
end
proteins.
of the neuron, at the end of the axon.
In motor neurons, the light chain of the Botox core molecule blocks the release of acetylcholine by cleaving SNAP25, which is an essential component of the snare complex.
So
When
that's what
acetylcholine
toxin
cannot
does.
be
It cleaves
released,
this
muscle
dominant
contraction cannot occur. In sensory neurons, the light chain is believed to cleave SNAP25 by a similar mechanism, thereby blocking the release of neuropeptide neurotransmitters and inhibiting the sensitization of pain nerves. The toxin does not appear to affect the conduction of electrical signals along the nerve fiber or the synthesis or storage of acetylcholine.
So did you hear that last part? So what does it do? It blocks the release of acetylcholine, right? It blocks the release of acetylcholine from the postsynaptic neuron, or the presynaptic neuron, excuse me. It does not affect the production of acetylcholine. It doesn't affect the activity of acetylcholine. It just prevents it from being released. So it stays on the presynaptic side in these membrane vesicles. But by blocking the release, right, it decreases overall muscle activity because you don't have the signal of the action potential crossing over. And so you don't have contraction that takes place. This is an example of a neurotoxin. Botulinum toxin is a neurotoxin. Now the effect is temporary, and you guys are going to brainstorm about this. Yes, question?
So in essence, in a way, it disables the nerves. In a way, it disables the nerves, so it kind of paralyzes them, and that's how it stops the wrinkles from happening?
That's right. So some of you are creating great opportunities for Botox as you wrinkle your brow right now. But you're wondering, is this Botox that I've heard people having injected into their face to get rid of wrinkles? Yes, that's what this is. But it's temporary. So how does it work? It does paralyze the nerve. Okay? It prevents the nerve from releasing acetylcholine to the other side. So another picture of this, a still shot, is showing this mechanism, botulinum toxin. You can see the full name in the upper right underneath their logo. Botox is a trademark, right? A registered trademark. And this molecule, the light chain of the molecule, there's a heavy chain and a light chain that the video talked about. But it's the light chain that cleaves a protein known as SNAP25. SNAP25 is a docking protein, or a protein that allows for the vesicles to dock with the membrane so that the vesicle can fuse with the membrane and dump out acetylcholine. So by cleaving SNAP25, these vesicles are not fusing with the membrane, therefore they're not releasing acetylcholine, therefore they're not crossing the cleft, therefore they're not binding to the other side. We know that acetylcholine is an ionotropic type of neurotransmitter. Remember that word? Ionotropic neurotransmitter is a neurotransmitter that directly influences the ion flow and the excitability of the other side of the... That's a bad spot for that. Right? An ionotropic neurotransmitter is one that binds and stimulates direct electrical changes on the other side. So if we talk about this a little bit, I want you guys to do this right now. Why does it work? I want you to be able to explain that to your neighbor. And then I want you to answer this question because I haven't explained why it's temporary, but it is temporary. In fact, if you know anybody that's had Botox, it doesn't last forever, right? They have to come back every 3 to 6 months and have new injections. Why? All right, why does it work? Yeah, you're hearing me talk about a cosmetic procedure and it totally works. There is science behind it. There's a lot of science behind it. Why does it work?
I'm thinking it's because since the nerves are more or less paralyzed for the most part, the skin doesn't move around as much, and so like it becomes tighter, and it gives the illusion that the wrinkles are disappearing.
That's pretty close, yeah. So it gives the appearance that the wrinkles are gone because the nerve is paralyzed, so it's not contracting the muscle. The muscle isn't contracting to pull on the skin. So the skin actually relaxes because the underlying muscle is not tugging on it. So, for example, there's another situation where you would see wrinkles disappear, but many times it disappears only on one side of the face. Those are stroke patients. Post-stroke, a patient on the left side, let's say the patient's 60 plus years of age, there'd be obvious wrinkles in certain spots as we get older. Okay, but on, let's say it was a right hemispheric stroke, the left side, the motor output on the left side would be paralyzed, so this side would be kind of droopy, and all of the wrinkles would be smoothed out because there wouldn't be underlying muscle tone. Okay? So we see the same kind of effect in nerve paralysis with stroke patients. So that's exactly why it's working. Now, why is it temporary? Who came up with that?
It's just like the other nerve will overcompensate for the fact that that's already like paralyzed, and the fact that the nerve ending can't grow back, it just has the like I can't answer it. It just decides to overcompensate for it.
So compensation. So let's pretend, that's a reasonable theory. Let's pretend that you injected enough Botox to take care of all the compensatory nerves in the area. Now what?
Does the SNAP25 get rebuilt over time?
It's a protein. What do you think?
I think yes.
I think you're right. So it's a protein, just like any other protein. It can be retranslated. As soon as it's retranslated and the protein is delivered to the end of the neuron, then now you have the ability for acetylcholine to be released again. Okay, and then you go back to the day spa and you get another injection, and then you look another 10 years younger. Okay? So it's temporary because the translation of the protein continues. So I'm just using this as an example to drive home a couple of key points at the level of the synapse. How it works, why it works, also why it exists. It's not so that we can look younger and not have wrinkles. It's so that we can modulate biologically what happens at that location. You can turn it up or turn it down. And you can even pharmacologically influence it, like what we do with Botox, what you do with cocaine, what you do with other analgesics like in surgery. All right, let's talk about different types of potentials. The neurons are going to integrate multiple signals. And these multiple signals happen throughout our day. They're happening right now. You're gathering information from all different areas that are surrounding you, and you summate these things. Some have said that the neuron acts like a democracy. Every vote counts. So every incoming signal is considered. If it doesn't reach threshold, we say it's a local potential or it's a graded potential. It doesn't become an action potential. Another question that came up in SI was the word dissipate. We know when we compare and contrast action potential versus graded potentials or local potentials will come up, but then they fade. They will go away. They will dissipate if they're not converted to an action potential. But an action potential is non-decremental, meaning it does not fade over time. Once it crosses threshold, when it comes down, as it sends its action potential to the next area, it's the same intensity every single time. So as we integrate these signals, we can have two main types. We can have an electrical signal that says stimulate the neuron, and we can have an electrical signal that says inhibit. If we say the electrical signal is excitatory or stimulatory, we call it an excitatory postsynaptic potential, otherwise known as an EPSP. An EPSP, excitatory postsynaptic potential. Or we could have one that's inhibitory. If it's inhibitory, an IPSP, then it causes more chloride ion to rush into the cell. If more chloride ion rushes into the cell, then, because chloride's high outside the cell, where sodium is, then the inside of the cell becomes more negative. And it becomes further away from threshold. So it's less likely to fire an action potential, therefore it's considered inhibitory. So an IPSP makes the inside of the cell more negative. And two neurotransmitters that would do that type of activity are known as glycine and GABA. In comparison, in contrast, or versus excitatory postsynaptic potentials, those are ones that cause sodium to come in, making the inside of the cell less negative, bringing it closer to threshold. And our examples here would be like glutamate and aspartate, as well as acetylcholine. You could add that to the list as well. Those are all considered excitatory postsynaptic potentials. Now, when we look at the different ways we can add these together, we can add them in two different fashions. We can add them either spatially over different locations, different neurons in space, spatially, are sending a signal. Or we can send a signal from the same neuron, but we can change the rate at which we send the signal, and we send it every couple of milliseconds. So temporally, we use the same location, but we send multiple signals over time. That's temporal summation. And spatial is multiple neurons in different locations over time. So let's look at examples of EPSPs and IPSPs. And we see this diagram. This is from a different textbook, but it's a pretty clear diagram. And this diagram is showing this postsynaptic cell. And here is an excitatory neuron, number 1, an excitatory neuron, number 2. This one's green, this one's black, this one's red, and that's an inhibitory neuron, number 1. Okay? You can see the plus because they're excitatory. You can see the minus because it's inhibitory. All three of these, and there could be others, are synapsing onto the same postsynaptic cell. So you have the ability to control it from 3 different locations. And this is a simplified view of what we have biologically. Well, if we look at what's going on here in panel A, here you can see the postsynaptic membrane potential in millivolts. You can see it's at resting about -70. And here's this little blip of a local potential. Local potential, excitatory potential number 1. And then it resets and fires again. Right? So same neuron over a different sequence in time. This would be a temporal summation, but it's not reaching threshold. Here's your threshold at -50 or -55, depending upon where you are in the body. The numbers are not exact. There's usually an average. Here you can see if we, over time, or temporally summate, and we fire the first excitatory from the green neuron, and before it dissipates or fades, we fire the second signal from the same excitatory neuron, we actually cross threshold and we fire an action potential, and this is temporal summation. Make sense? Let's look over here at spatial summation. Well, in spatial summation, you can see the effects of temporally summating, but not close enough, excitatory neuron 1 and 2, just like what we saw over here in panel A. But here, we actually fire excitatory 1 and 2 right on top of each other. So EX1 is the green one, EX2 is the black one, and those summate to cross threshold and cause an action potential. So this would be an example of spatial summation. Why? Because these two neurons are in different locations. Temporal is the same neuron, back to back. Spatial is 2 different or more than one simultaneously before they dissipate, so that you actually reach an action potential. And then let's not ignore the inhibitory one. So over here in panel D, here we've got an EPSP minus an IPSP, otherwise known as cancellation. So in this situation, if you had an excitatory, the green one fires, and then you follow it with an inhibitory one, over time, nothing happens. But if you summate them together, they cancel out. You could also appreciate if I had a panel E, for example, I could actually fire 2 of the inhibitory one back to back, and I could take this down to like minus 90. You with me? So this gives you an explanation of the differences between EPSPs and IPSPs, but also temporal and spatial summation.
So if you fire the same one, like in box A, nothing happens, they have to be different?
Well, no, they don't have to be different because in box B they're the same. The question is, if I fire the same excitatory in box A, they don't reach threshold. But they do in box B. So what's different between A and B? What's that? A is local and B would be considered an action potential. How did you get to an action potential? Because look at the difference in space in time. This is milliseconds. The gap between these two arrows, and look at the gap between those two arrows. So the panel B, the gap between the two arrows is closer together, less of a gap, so that means they're closer in time. So in order for it to be temporally summated, the time has to be short enough that you fire the next one before the first one fades off. Okay? Yes?
So I have 2 questions. So number 1, it looks like from what you're describing that there are like big neurons that are in charge of transmitting signals, and there's like tiny neurons that their only job is to either inhibit or excite the big neurons. So there's like 2 different kinds of neurons. There's the big ones that actually do the communicating, and there's the little ones that either excite or inhibit them.
No, all neurons are either excitatory or inhibitory.
Okay.
And their relative size really only influences the speed of conduction.
Okay.
So a larger diameter neuron will actually travel an action potential faster than a smaller diameter neuron.
Okay, so what you're trying to say is that the neurons, they can all inhibit and excite each other.
Yes, as long as they're wired for that. There are certain neurotransmitters at the end in certain vesicles that will be released, whether it's inhibitory or whether it's excitatory. So this is a good question. If you have a network of neurons, like a road map in the body, you don't want certain roads that only allow you to turn right. It's nice if you have a road where you can go right or left. Okay? You want neurons that can be inhibitory or excitatory. The way you control whether it's inhibitory or excitatory is the type of neurotransmitter you release at the end of the neuron. So you use the same biological architecture, you just modulate at the end whether you're turning right or left. Okay?
One last question.
Okay.
I remember when you showed us how the dopamine was coming out of the neuron and then how it would go back in. Does that mean that every neuron has every single type of receptor, dopamine, acetylcholine, serotonin? Does that mean that every single neuron has all the different neurotransmitters?
No, they have. So the question is, does every neuron have all the 100 plus known neurotransmitters? No. There are certain neurotransmitters that are really isolated in the central nervous system. There are also certain neurotransmitters like the ones that are tied to the limbic system on emotion. Right? Things that actually get you excited, emotionally worked up, or emotionally depressed. Those happen within the limbic system, and those neurotransmitters are unique to the limbic system.
Okay, what I'm trying to say is, would every single neuron that specializes in, like you said, like there's the ones in the central nervous system, like would every single neuron in the central nervous system have the specific neurotransmitters that they deal with stored inside them all the time? Because the way you described it, it would go back into the neuron, but wouldn't that happen every single time? So where is the neurotransmitter?
The neurotransmitters are stored in the presynaptic side in vesicles, in little compartments. And they're stored there. If they have the capability to make that neurotransmitter, they're stored in the end bulb.
Oh, okay.
Okay.
That may be a problem with people with certain mental illnesses, and maybe they don't have the correct neurotransmitters inside.
That's absolutely correct. In fact, serotonin is one that's really been tied to depression. Okay? So that's way beyond this class, but you guys will get into those things. So let's talk a little bit about this diagram. This is another way to look at, these are good questions, guys. I have to keep it going, okay? I'll try to handle as many of them as I can, but once I feel like we're going to get stonewalled, then we'll have to kind of move on. The EPSPs that are shown here in these arrowheads, there's 3 of them. Okay, and these were like chicken scratch drawings by myself, right? So that's why they don't look all smooth, and and nice and clean like some of the other diagrams that we've seen. But if you see that these summated, right, they summate to a minus 50 or minus 55, triggering action potential, then it comes up. And this would be 3 EPSPs summated over time to actually trigger an action potential. Well, when we integrate it all over the place. And we've got a couple of different ways that we do this. When we look at temporal summation and spatial summation in just a different figure, so it's the same information now presented in a different figure. Right? Here is temporal summation, so you can see that there are 3 individual neurons all converging on one neuron. But the middle neuron is the only one that's firing. You see these little tick marks? Those are triggering EPSPs. Those are local graded potentials. So now we have 3 or 4 terminologies for the same thing. A local or a graded potential is not an action potential. It could be either excitatory, so it's an excitatory postsynaptic potential, or it could be inhibitory, which is an inhibitory postsynaptic potential. Here we've got these hash marks indicating these EPSPs, and they spread from one synapse to the trigger zone, and then they cause the postsynaptic neuron to fire. Because these temporally, over time, summate like on that previous diagram. Or spatially, you can see that there's more and they're closer together, meaning shorter in time. Or here you can see that there's fewer, but they're spatially from 3 separate neurons. And those added together, summated, that's what that word means, at the trigger zone allows for a postsynaptic neuron to fire. So here is another way to look at the same concepts of spatial versus temporal summation. Now, as we wrap up this particular lecture, the last slide I want to leave you with, that we're going to come back to on some of the different motifs as we cover the nervous system, is looking at the different types of circuitry that exists. The circuitry that exists usually falls into one of these 4 categories. We can have circuitry that actually is an input coming in, and it diverges to many different outputs. So those are called converging circuits. As we organize our neurons, we have one input coming in with many, many outputs. This is actually good circuitry for large-scale muscle contraction. So think about your quadriceps or your hamstring muscle groups. You want to be able to fire them all at the same time, and you only want to have one signal that goes to the muscle group telling it what to do. So you use diverging circuitry for motor output. Now, converging circuitry, the upper right, what do you think this motif would be wisely used for in the body? Sensory. Okay, so sensory, this is very, very valuable for incoming sensory information where it converges on one part of the brain. So if you think about the input, right? Think about when you're walking in your bedroom at night trying to find the bathroom. You know, the lights are out, right? So you're getting input, sensory input, right? And then you're like, oh, there's the table, right? Because you didn't notice that it was going to hit you in the shin. So you have sensory input from many different places converging into the brain to detect what's going on in the central nervous system. Okay, these 2 are a little bit more unique. These are the main ones. These 2 are also present, but to a lower extent. A reverberating circuitry that's shown here is the promotion of the input stimulating an output, but before it does that, it comes back and triggers this to happen again. So I want all of you to practice this activity. Ready? Take a deep breath. That's reverberating circuitry that allows you to continue to contract the diaphragm so that it compresses down and you continue to enlarge the thoracic cavity to take in a deep breath of air. So the reverberating circuitry is important. It stops once you exhale because you don't need that happening anymore. Parallel after discharge. So this one is, you've got parallel circuitry. You have inputs coming in, and you have parallel circuitry leading to an output. And this happens within the circuitry of the eye. So if we did this a little experiment, but we won't, but you've probably seen this happen. You could stare sort of at one of these bright lights for a period of time. Okay, and then you could close your eyes and you would still see an image of that light bulb or that bright spot. Or you might even still see it as you look around the room.
I usually see a red spot, like a little white light.
Or a red spot, okay? So that's a parallel after discharge that allows you to see that light bulb after you've closed your eyes or after you've looked away from where that image actually comes from.
Is that how our illusions work?
Illusions? Like, you talking like David Copperfield kind of illusions? Optical illusions? So optical illusions usually are explained by sort of the angle at which you're looking at the object, not necessarily circuitry. Okay? All right, questions over the nerve physiology segment?